First of all, I would like to thank Dimple, Ranjith, and Mithun for giving me opportunity to present our work here. And unlike Aja's excavation of nucleosomes, in our lab, we are building the heterochromatin to understand the epigenetic mechanisms. In a broader perspective, in our lab, we are trying to understand how different cell types are derived in our body. And most importantly, when we look at in the human body, around more than 200 types of cells are produced over the lifetime. So, however, all of these cell types carry identical genome. So how this is possible? So this is mainly achieved by differential gene expression. And for example, if we take the neuronal cells, only neuronal specific genes are activated, and similarly, the non-neuronal genes are inhibited by transcriptional factors, also by epigenetic mechanisms. So as I mentioned, generally by these cell type specific enhancers are bound by the cell type specific transcriptional factors, and they recruit different cofactors. Either they activate the gene or repress the gene based on its nature. Also, they at the site, either they incorporate active histone modifications or repressive histone modifications. So in the case of especially in the case of the repressive histone modifications, we see that for example, H3K9 methylation or H3K27 methylation and DNA methylation, even these are after the differentiation, even in the absence of the sequence, even in the absence of the sequence-specific factors, these heterochromatic marks are epigenetically propagated through the multiple cell divisions. So we wanted to ask how what are the sequence of events during the establishment of these heterochromatin domains, and what are the kinetics, and what is the contribution of these histone modifications and DNA methylation in maintenance of this propagation, maintenance of epigenetic memory. So for this, we designed an assay called chromatin in vivo assay. So we designed a reductionist biosynthetic approach called as a chromatin in vivo assay. So basically it's a simple a GAP reporter driven by a constitutive strong promoter, and upstream to the promoter we inserted a 7x stato binding sites. So the idea is, for example, a protein like HP1, if you fuse to this bacterial tetrapressor and introduce into the cells, this because of its affinity to teto binding sites, it binds there and it recruits the H3K9 methyl transferase, and thereby it initiates the heterochromatin. Also, and thereby we can, at our will, we can initiate the establishment of the heterochromatin, also the establishment of the broader domain of the heterochromatin. And after that, if we want to test the epigenetic memory by adding the doxycycline, we can release this to that HP1, and then we can ask if it is maintained in a epigenetic manner or not. So for this reporter, to insert this reporter, like in the genome, we could have inserted this reporter in multiple, like thousands of sites, but we choose the OCT4 locus as a region to insert this reporter. The main reason is OCT4, as we know, it's a pluripotency factor, and this OCT4 is controlled by an upstream enhancer. So, and we know from the literature that in the somatic, after the differentiation in the somatic cells, the OCT4 is silenced by H3K9 and DNA methylation. And this H3K9 methylation is initiated by G9H-GLP. But however, we don't know how this G9H-GLP is targeted to this OCT4 locus. So therefore, if it is since it is already a target of H3K9 methylation, so what we did is we replaced the one exon of the OCT4 with the GAP with the poly A, and in between the enhancer and the promoter, we inserted the 7x strato binding sites. And since the OCT4 is a haplosufficient gene, so even if we perturb or disrupt the one allele, the pluripotency of the cells is not affected. And into these OCT4 reporter cells, you can see that This GFP is driven by ARC4 promoter, and it is this is the FACS data. We can see that the GFP is nicely expressed. And into these cells, we introduced the HP1 fused to the tetrapressor, and we saw that there is a robust silencing of the reporter highs happening in these cells. And most importantly, you can see that if you introduce the bacterial tetrapressor as a control, you see that it has absolutely no influence on the reporter expression. So then what we did is we, in a timely manner, I'm showing the one time point here. So here we did a chip qPCR to check the different histone modifications. For example, in the case of the control recruitment, you can see that active marker, the HTK4 trimethylation is quite nicely enriched at this reporter locus, whereas we saw insignificant enrichment of the silencing marks. But after the recruitment of the HP1, you can see that the HTK, the active marks are disappears from this region, and only the silencing marks are enriched there. And also we checked at the DNA methylation. You can see that in the case of the control, there is absolutely very little or insignificant DNA methylation is there. But in a time-dependent manner, the DNA methylation starts accumulating at this reporter locus. So taken together, so we shown that just like in the cells, recruitment of the HP1, it establishes the HTK9, also DNA methylation at the reporter locus. So by performing qPCR and DNA methylation analysis in a timely manner, we found that these HTK9 methylation domains are established in a symmetric fashion, 5 kilobases either side of the reporter nucleation site. Also, we calculated that around 0.18 nucleosomes, the spreading rate is around 0.18 nucleosomes per hour. So then we asked whether this established heterochromatin domain is stable enough to survive the replication stress or the replication dilution. So here, then by adding the doxycycline, and after the 4 days, which is approximately about the 8 cell divisions, we analyzed, and you can see that around major or the 96% of the cells maintain the reporter silencing. Not only the reporter silencing, we also saw the maintenance of the HTK9 methylation, also the DNA methylation. So therefore, with this assay, we could show that we calculate the kinetics of the spreading rate of the heterochromatin, and also what kind of, how big the sizes of the heterochromatin domains established. Also, we show that there is the establishment of HTK9 methylation and DNA methylation at the reporter locus. And this suggested that maybe there is a positive feedback loop, and that is what helping in the maintenance of this heterochromatin at the ARC4 locus. So to test this, first we depleted the HTK9 methylation, but however, we find that HTK9 methylation is absolutely essential for the initiation of silencing. And then we depleted we made a knockout of the DNMT1, which is the maintenance methyltransferase. And you can see that in the case of the DNMT knockout, the establishment of silencing happens normally. We see the establishment of HTK9 methylation. Also, there is a silencing of the reporter. But however, when we test for the epigenetic memory, you can see that the reporter is activated within say 8 cell divisions or within the 4 days. So with this, we proposed, from this data, we found that the HTK9 methylations or the domains spread around 0.18 nucleosomes per hour. And this domain, the HTK9 domains are formed around 10 kb symmetrically from the nucleation side. And we find that there is a positive feedback is driving the epigenetic memory in the case of the mammalian cells. So then we asked, what are the other cofactor, or what are the factors which are recognizing HTK9 methylation and driving the DNA methylation? And from the DNA methylation, what is again feedbacking or the enhancing the HTK9 methylation? So of course, from the literature, we know some, and we thought that by performing a genetic screen, we could identify maybe novel factors. So for this, we performed a genome-wide CRISPR-Cas9 screen. So what we did is we took a silenced reporter cells which are expressing stably Cas9, and into this we introduced the sgRNA library. And after the selection, we also added the doxycycline so that we will also able to find the maintenance factors. And around 1% of the cells, we saw activation of the GAP. And these GAP activated cells, we fact sorted, and by sequencing the guide RNA sequence, we identified the genes which are playing role in this maintenance of the, or initiation of the heterochromatin, also the maintenance of heterochromatin. And as expected, we find the DNMT1 and UHRF1, which are the maintenance methyl transferases. Also, we find HTK9 methyl transferases. And in addition to that, we find few novel factors, and among them, this zinc finger 462 caught our attention because it is a sequence-specific factor. So what is a sequence-specific factor is doing in the maintenance of this epigenetic memory at this reporter? So what we did is we did the multiple validation studies, and after that we also performed the chip sequencing for zinc finger 462, and we find that zinc finger 462 binds at the enhancer region of the OCT4, where the OCT4, SOX2, and NANOG is binding. And by biochemical studies like protein-protein interaction studies and other experiments, we find that zinc finger 462 by interacts with the G9 GLP, and thereby it recruits the HTK9 methylation at the reporter locus, and the established HTK9 methylation promotes the DNA methylation, which is later epigenetically maintained in the differentiated cells. Of course, here in the embryonic stem cells, of course, the zinc finger 462 is required because this enhancer is constantly bound by activators, so there is a competition going on. But when we tested this requirement of zinc finger 462 for the epigenetic memory in the differentiated cells like MEFs, we find that zinc finger 462 is not required, and this HTK9 methylation and DNA methylation is sufficient to drive this epigenetic memory. So then we asked zinc finger 462, is it regulating only OCT4 or also it is regulating other genes in the genome? Since we have the ChIP-seq data, so we analyze this and we find that in addition to OCT4, zinc finger 462 also binds at around 16,000 genes in the genome and 16,000 regions in the genome. And by analysis, we find that all these 16,000 regions are mesoendoderm specific enhancers. So I'm not going through all the data, but what we find is zinc finger 462, by recruiting the G9 GLP at the mesoendoderm specific enhancers, it silences the mesoendodermal genes during the neuronal specification, thereby it gives the specificity or robustness for the neuronal differentiation. So this was mainly, with this, we shown that HTK9 methylation indeed we identified the sequence of events. Also, we show that HTK9 methylation and DNA methylation, there is a positive feedback loop which drives the epigenetic memory at the OCT4 locus and possibly in the other regions of the other target regions. So then we went ahead to characterize or understand the epigenetic mechanism of other kind of epigenetic mechanism, which is polycomb silencing. As on Tuesday, Professor Rakesh mentioned, from the flies to animal vertebrates, Hox genes control the organs development. And this differential expression of these Hox genes is regulated by polycomb proteins and mainly called as the polycomb repressive complex PRC1 and PRC2. So PRC1 catalyzes the H2A113 ubiquitination, and the PRC2 catalyzes the HTK27 trimethylation. And it is believed that the ubiquitination promotes the HTK9 methylation, and HTK27 methylation promotes the ubiquitination. Thereby, it is the epigenetic memory is maintained at these target genes. But in 2017, there are multiple studies came suggesting that in addition, for the maintenance of this silencing, DNA sequences or the specific elements are required. So this is put the question on this epigenetic memory by polycomb silencing. Therefore, to address this, we wanted to use our chromatin in vivo assay, and thereby can we address whether the stone modification is sufficient to maintain the epigenetic memory of the polycomb genes. So not only that, in addition to that, in case of the vertebrates or in the mammals, the PRC1 complexity escalates. For example, we find a canonical PRC1, also variant PRC1 in the case of the mammals. And in addition to that, if you consider the PCGF types, this further complicates. So in total, we find 2 types of canonical PRC1, and there are 4 types of variant PRC1. So, okay, leaving this apart, so first thing we wanted to ask is, what is the contribution of this canonical PRC1 and variant PRC1 in maintenance of the establishment and maintenance of the polycomb silencing? So for that, what we did is, since the OCT4 is under the influence of HTK9 methylation, so we created a reporter line by integrating our reporter construct cassette in the intergenic region where the histone modifications are not observed. It is kind of, we call this as a naive reporter. So into this one, we introduced the variant PRC1, also canonical PRC1, and you can see that by recruitment of the variant PRC1 or the canonical PRC1, both of them are able to efficiently silence the reporter. But however, when we test for the epigenetic memory capacity for these 2 complexes, we see that in the case of the variant PRC1, the silencing is demolished at the reporter site, but in the case of canonical PRC1, you see that the silencing is maintained. So what is the difference between variant PRC1 and canonical PRC1? So if we look here, the canonical PRC1 has a CBX1, which is the reader for the HTK27 methylation. But in the case of the variant PRC1, it don't have any reader for the HTK27 trimethylation. So then we thought that maybe canonical PRC1 is able to establish a feedback loop with the PRC2. Therefore, the canonical PRC1 is able to maintain the epigenetic memory, but variant PRC1 fails to maintain the epigenetic memory. To test this one, we made a catalytic dead mutant of CBX7, and then we introduced into the recruited at the reporter locus, and we find that whether the wild type or the mutant one, when we recruit, we saw the establishment of the heterochroma polycomb silencing marks. But however, when we tested for the epigenetic memory, you see that the reporter is activated. Also, I'm not showing the data, these polycomb silencing marks are erased from the reporter locus. So suggesting that, so indeed the CBX7, because of its having a reader binding pocket for the HTK27, it is able to establish the polycomb feedback loop with the PRC2. Therefore, this it establishes a stable heterochromatin domain, and thereby it is epigenetically maintained during the cell divisions. So this is with this by using our biosynthetic approach called as the CIA, we were able to show that the sequence of events, which I didn't mention about the PRC2 or the polycomb. So we could show that the kinetics, also about indeed the histone modifications and the DNA modifications are sufficient to maintain the epigenetic memory at the target sites. So in addition to that, since our reporter, I'm going to present, which is not related to the epigenetic memory, but here, since our reporter is very sensitive for the any transcriptional or any factor, if we recruit there, if it is a repressor, it represses the reporter. Also, if it is the activator, it activates the GAP expression. So therefore, we thought that why not we use our CAA reporter approach to validate or maybe test all the genome-wide, all the proteins, their capacity to activate or repress. So for this, initially we planned to clone the whole genome cDNA with the tetrapressor, and then we thought that by introducing one by one in a bigger screen, we could identify the silencer proteins across the genome. But however, while cloning the cDNA, we realized that it is very difficult, challenging, because we attempted once or twice, only the small fragments, smaller cDNAs we were able to clone, but the bigger ones we failed to clone. In the complexity of this cDNA library was very low. So to overcome that, we thought that why not we hack our splicing machinery, thereby we can tag the entire proteome, and by performing this tagging inside the reporter cells, we could screen for the repressors or activators. So here, for this, we designed a retroviral cassette. You can see we call this as a ORF tag. So in this one, it has a neomycin cassette, followed by iris and a tetrapressor. And to the tetrapressor, we fused a strong splice donor. And this complete cassette is completely driven by a strong promoter. And we know that when we introduce this into the cells, most of the time these retrovirus elements integrate near the promoter regions, either in the promoter regions or maybe in the near the promoter regions. So let us say if it is false in the intron 1, so since this is having a strong splice donor, it attacks the downstream exon, and thereby it makes a chimeric construct which is fused to the tetrapressor. So if we perform this in a huge number of cells, so making sure that each cell gets a single this ORF tag cassette or the construct, thereby actually this works extremely well. So where we perform this screen to identify the repressors, and around 1% of the population, we started the screen with the 500 million cells, and from that around 1% of cells were showing the reporter silencing. So we fact sorted that, and by NGS sequencing, we identified the genes which are causing the repression. So with this ORF tag approach, we could tag, you can see that around 85% or the 85% of the genes we could tag with this ORF tag approach. And we, from the beginning, we know that the intron-less genes, we are not able to tag with this ORF tag approach because they don't have any exons. That means they don't have any, there is no splicing happening there. So what we did is we analyzed what are these intron-less genes, and we find that these are either interleukin related or receptor proteins or the histone proteins. So by looking at this, we were sure that we won't miss out any genes which are, maybe we won't miss out majority of the genes which might be playing role in the transcriptional repression. So with this, here is the data. So in the sorted population, we saw the enrichment of about 300 proteins. And from that, most of the, around 100 proteins are crab zinc fingers. Also, I highlighted some of the already well-characterized heterochromatin or the silencing proteins in this plot. And here, this is how the data looks. You can see that this is the unsorted population. It is one of the top hit. This is ZFE 57. You see that the retroviral integrations in the unsorted population, we see uniformly distributed in the sense and nonsense strand. But however, when in the sorted population, since the gene is on the place strand, you see that the integrations are enriching towards the only place strand. So with this approach, not only for the silencing, our collaborators perform for the activators, also post-transcriptional regulators. And this approach gave us excellent results to identify the novel factors regulating either the transcriptional regulation, also the post-transcriptional regulation. So with this, recently, all this work, what I shown is done in I was a joint postdoc at the Oliver Bell and Julius Brennicke's lab at Vienna Biocenter, and all this work was done in there. And last year, we started a lab in CDFD, and this is the status of our current lab, and we are trying to understand whatever these transcriptional repressors we identified, we are trying to understand if they play a role in the lineage specification, and that's what we are going to do for the next few years. And thank you all.
Thank you, Ramesh. We'll take 3 questions.
Hi, very nice talk, very interesting. The first part you used the reporter line for HP1.
Yes.
So I wonder if you target that to a different genomic locus, for example, transposable elements, would you get a different type of...
So what frankly, I tried it at 30 different locuses in the genome, and we didn't try in the transposable loci because transposable element loci, it is expected to silence already. So, but we tried it around 28 intergenic regions which are not having any silencing or active mark, but surprisingly none of them were showing the maintenance except the OCT4 locus. So what we find later was some reason OCT4 locus is resistant for the TET enzymes, but all other regions, the TET enzymes were, as soon as the DNA methylation is established, it is erasing them. So by deleting the TET enzymes, then only we can see the maintenance in other loci.
Thank you.
Hi. The Orpheum tagging is really clever. When you get the hits for the repressor,
Yes.
and I presume you sequence those cells in some sort of, so you can identify insertion sites.
We are using IPCR followed by the, yeah.
So can you see roughly where the TET repressor is in those fusion proteins?
Yes, it means basically IPCR, how it works is our primers are in the LTRs.
Yeah.
So therefore, we know the direction. So therefore, we can guess where the tetrapressor is in these fusions.
And is it mostly at the N terminus?
This is always, most of the time, because we know from the, previously we did in our campus, we did multiple screens. So the retrovirus mostly goes to the transcriptional start sites. So it is a more or less, I would say, N-terminal tagging.
Which virus is it?
This is retrovirus. We also did with the lenti and the piggyback. Even lenti, I would say it may go to the more or less little bit, not as biased as the retrovirus. But what we find is, for example, instead of, if you want to tag the C-terminus, you replace the splice donor with the splice acceptor, then you can tag it to the C-terminus.
That's very clever.
This corner, Ramesh. I wonder if you have seen, if you have checked this repression that is established by HP1, and of course it is synthetic, for how many generations it stays?
Okay, so whatever the data I shown here is the 8 cell generations, but we kept the cells for almost 30 days. I would say it is stably maintained up to 40 generations. But it also depends on how much time we are recruiting the HP1. For example, if we recruit the HP1 for, let us say, a month, and after that, if you are untethering by adding the doxycycline, the stability is even longer because the accumulation of DNA methylation is quite higher at this loci. So it's purely the maintenance of the memory depends purely on how much DNA methylation is accumulated at the reporter locus.
Yeah. Do you think when you allow this recruitment of HP1 for longer periods,
Yes.
it leads to the longer and wider spread of this domain?
So I agree, but it also it depends on what kind of genes present in neighboring regions. Also, the CTCF binding sites might also play a role. So here we only did after 21 days, and in that one we find that the domain what is forming is up to 5 kilobases on the either side, and it is symmetric. And of course, if we would have done longer time, maybe we would have seen how long it goes, we don't know. Yes.
Thank you.
We'll take one more question.
A lot of data, thanks. So I was wondering, you mentioned about G9A and GLP, right? Yes. I was thinking, how did they do the DNA methylation?
It's not DNA methyl, G9A, GLP are the H3K9 methyl transferases.
Yes.
So, of course, once the H3K9 methylation is established,
yes,
we suspect that since we are recruiting the HP1 at the locus, the HP1 not only reads and oligomerizes, it also has the capacity to recruit the H3K9 methyl transferases, like CDB1. And we suspect that the CDB1 might be recruited and it establishes H3K9 methylation, and the H3K9 methylation is read by HP1, and wherever the H3K9 methylation is there, UHRF1 is known to bind, and that's why DNMT proteins might be recruited. So this is more or less according to the literature, but I would say maybe it's not fully understood.
Okay, thanks.
Maybe last question, and then we'll part for tea.
So when you recruit HP1, can you see a localization of your locus close to, I don't know,
this is actually
pericentromeric regions or whatever?
We expect that after the heterochromatization, we suspect that it is relocalizing to the nuclear periphery. And actually, in our screen, we got some hits which are localized to nuclear surface. But I got busy with this zinc finger 462 other factors. We did not pursue them, but I think currently, right now, we have time, so we are going to do that. And we also try to image before the silencing and after the silencing by making an array of CRISPR array. But initially it was very tough. I think maybe with the improved technologies currently it might be possible to image before silencing and maybe dynamics of how it is moves and tethers to the nuclear membrane. Yes.
Thank you, Ramesh.
Thank you.